Well, hello everyone. Everyone at home watching. I'm going to review the integumentary system. Your exam one is due at the end of the week. Today is the Thursday, the 25th, so it will be due by Sunday. So keep that in mind. But anyhow, let's just review the integumentary system, and this should be the last chapter that's going to be part of exam one. So it's going to be exam one, chapters 1 through 5. So here is a view of the integumentary system that we went over it on Tuesday. Here you have your epidermis with several layers, 5 layers present within the epidermis. 5 layers present in the epidermis in thick skin, the skin in the palms of your hands and the soles of your feet. Now, if you have on the rest of your body, you're going to be missing the stratum lucidum, this clear layer of epithelium underneath the stratum corneum. It'll be like the second layer from the apical side, from the exterior. We're going to be missing that in most of our bodies. So that is why most of your body will contain 4 layers of epidermis, 4 layers, an epidermis with 4 layers, whereas your thick and thin skin will contain 5 layers, the extra layer being the stratum lucidum. So what's happening within the epidermis? We're gonna go over it in more detail, but here we have a basal layer. We talk about the epidermis doesn't have any vascularization, and just like any other epidermal tissue. And the vascularization is provided by the underlying tissue. In this case, it's usually connective tissue. In this case, we have areolar connective tissue in the dermis, on this dermal layer. And this is where you're going to have a vascularization that is going to provide nutrients and oxygen to the epidermal layer. So here we have we went over the epidermis, and now underneath the epidermis we have our dermis with a papillary layer and a reticular layer. These 2 layers will contain vascularization and nervous innervation. Your epidermis itself will also contain will contain Merkel cells, which are going to be there for innervation as well. Usually, whenever you think about this sensory, your senses, they're usually comprised of nerves. I reckon that's something that we all kind of have an intuition. Now, where are the cell bodies of those nerves? So usually the layout that we will have, and we will see it on detail whenever we look at our nervous system, the cell bodies of those nerves, they're going to be in these collections of cell bodies parallel to your spinal cord called ganglia. So there's just going to be collection of neuronal cell bodies running parallel along each side of the spinal cord. And these cell bodies of these neurons will project their axons towards every single millimeter of your skin in order to get that sensory signal. Now, so the answer to that is the cell bodies are in the ganglia that are parallel to the spinal cord. Now, there are these nerves, sometimes they end up as free nerve endings, but for the most part, your sensory system, be it your skin, be it your the olfactory, be it your gustatory system, they will have the endings of those neurons, the endings of these axons will have a specialized cell that surrounds it. And the specialized cells, think of it as a little antenna that captures whatever signal it's interested on. In this case, we're interested on this signal of the sensation, this signal. So there's going to be a specialized cell next to the nerve endings that whenever you feel any sort of sensation, there is contact with your skin, the specialized cell will send a signal to these free nerve endings, and the free nerve endings will in turn send the signal to the ganglia that are parallel to your vertebra, and from there to the spinal cord, to higher centers. So those here we'll see in detail later the presence of these Merkel cells, and that serve this role as an antenna, as a mediator of the signal, sending the signal to the nerves that eventually send it to the cell bodies of these neurons. Okay, so what else do we see here? We see the accessory structures that we have present on this on your cells. You have here you have here hairs with adjacent usually sebaceous glands. You have here sweat glands for thermoregulation. And underneath you have the subcutaneous layer composed mainly of adipose tissue. Here, this is highlighting the subpapillary plexus. We mentioned what is a plexus. It's a network. In this case, you'll hear the name plexus associated with a network of blood vessels or a network of nerves. In this case, we are talking about a network of nerves. So here we have the subpapillary, so under the papillary layer, there is this network of nerves, of blood vessels. What else do we have here? So here you have pores for your sweat glands, your hair shaft, your hairs are usually gonna be associated with a sebaceous gland. Sebaceous gland which releases sebum, this oily substance that's there to coat to produce an oily coat to this hair, to the hair shaft. Now you can see that would be beneficial if you are if it rains and you have a layer of and you know, our primates, our ancestors, and most animals out there, they have, or all mammals, they have a layer of hair, and this layer of hair, it's coated with this oily substance so that whenever it rains, the rain doesn't infiltrate into deeper layers. So it's just like we're somewhat impermeable because of this coat, and that produces that advantage that we won't lose as much heat due to the presence of rain. Here we talked about erector pili. We talked about the presence of lamellar corpuscles and these different layers that allow it to sense pressure. Tactile corpuscles. Here you have these arteries and veins present within the cutaneous plexus, and this.
I have a question, or maybe two.
Yes. Go ahead.
So the hair, when sometimes they fall off naturally, and it comes out like the whole follicle with the little the bottom part of it, the root.
Yeah.
Does it, when that happens, does the erector pili comes out too, or does it stay? It stays, right? And attaches again to a new hair?
Yeah, I've reckoned that is what happens. The erector pili stays, so that infrastructure, the erector pili and the connective tissue that it's attached to, that remains once we lose our hair. And I reckon we can try it, right? Pull a hair and then kind of have a label on it, and by the end of the semester, see if it comes back. It should. It should come back, and it should stand up because the erector pili would still be present.
Okay, thank you.
All right. Give me 1 minute, please. Sweet, I'm back. Okay, let's look at this. So here's just a big picture overview of what we're looking at. Your epidermis with a papillary and a reticular layer. You're going to have a level of innervation at the epidermis as well as the reticular layer that's underneath it. Well, there's innervation at all levels, but in terms of what you think about pressure and touch sensation, they're going to be interspersed between both layers, epidermis and dermis. The epidermis itself, we mentioned it's responsible, it plays a role in the synthesis of vitamin D3. The papillary layer, the one that's immediately adjacent to the epidermis, that's the one that's going to have the most vascularized area in order to supply this highly proliferative basal stratum where the stem cells of the epidermis reside, basically. So when you think about it, this layer right here, this is your epidermis right here. It looks like two layers because there's a lot of stem cells here at the epidermal ridge, at the basal stratum, and there are stem cells here, and they're going to divide, produce these melanocytes, keratinocytes, and those are going to divide and make a lot of keratin, and eventually their organelles are going to wither, and they're going to die, and they're going to create this layer that's mainly filled of cells that they're nothing but keratin. So that's why it looks like 2 layers, but this is, you can think of it, this is where cell division happens, and as they differentiate, as they go towards the surface, they start dying off. And that's what's being depicted here. And those are the 5 layers that we mentioned. This is on the epidermis, this is the stratum corneum. But one thing about this epidermis is that there is no vascularization there. So it has to come from the dermis. And here you see this capillary network being close to your epidermis, because this is where oxygen is going to come from. This is where nutrients, glucose to provide energy for cell division, that's where it's going to come from. All right, so here we were talking about the papillary layer. The papillary layer providing all of those nutrients. Then we have a reticular layer under the papillary layer, still dermis, containing all of these receptors, all of those nerve receptors for pressure, for sensation, tactile receptors, pressure receptors, sensitive receptors, and all of the blood vessels and that supply the epidermis. The hypodermis is not mentioned here, but it is for the most part named as a portion of the integumentary system. The hypodermis being that layer of fat that if you ever get a deep cut, you will notice that layer of fat. Here are the accessory structures present. We talk about hair follicles, exocrine glands. They are going to be releasing water, and it's going to release whenever you sweat. You notice that your sweat is salty, so you're going to have ions present within your sweat. And you have also fingertips, which are another accessory structure. Hair and fingertips, and the stratum corneum, the most superficial layer of your skin, they're all going to be made from keratin. They're all going to have that keratin as their main component, this protein that just forms these fibers. And depending on the density and conformation of these fibers, that's going to determine the rigidity and thickness of it. So this is, here is a view of your epidermis. I reckon this is a view that allows you to recognize this epidermal ridge that is complementary to the dermal papilla. We talked about this being, these ridges and this papilla being the reason why we notice our fingerprints. They say that no two are alike, but there have been cases in which people have been misidentified because of their fingerprints. We do have fingerprint twins. It's just a matter of whenever you have an event occurring so many times, then there is the likelihood that they will be the same. That, I bet that also applies for snowflakes. So it's just, uniqueness is rare whenever you have an event that repeats itself so many times. All right, so here we have cells of the epidermis, keratinocytes, thin, thick skin, difference between thin and thick skin. Thick skin, palms of your hands and feet, and thick skin contains an extra layer. That's going to be your stratum lucidum, and we're going to see those layers. So here they are. Stratum corneum, the layer of dead or dying So let's start from the bottom. So this is your epidermis present here. You start with the stratum basale, and this is going to be the stratum that contains most of your stem cells, your cells that are undergoing cell division. And it's going to be your basal cells, column stem cells, column basal cells. But just know that this is where cell division occurs. And this is a repeating pattern for epithelium, all epithelium. There's a layer um, adjacent to the basement membrane that has mitotic capacity. Then you have a stratum spinosum. The stratum spinosum takes its name due to the presence of keratinocytes bound together by desmosomes. Now, what does that mean? You have to know what a desmosome is, right? Does anybody want to take a stab at a desmosome? So when we were looking at epithelium, we look at the polarity of epithelium, and we mentioned several components that allow cells to form these epitheliums, to form these contiguous or connections between epithelial layers, epithelial cells, or between the epithelium and the basement membrane or basal lamina that's underlying the epithelium. In order to do this, there's gotta be points of junction, right? Points where the cells are going to touch each other, or where in the case whenever they're adjacent to each other, or whenever the cells are going to touch the underlying layer, where cells are going to touch, in this case, this layer over here, where the epidermis meets the dermis. This is done through these anchoring proteins that interlock with each other and are contiguous, or are points of attachment to the cytoskeleton, intermediate filaments of the cytoskeleton. So that is, so let's say if an analogy, if we humans were to form like these contiguous structures next to each other, and we wanted to just like have that real form a wall, an impervious wall, it would be analogous to us fusing with the person next to each other at the having proteins here that allows us to fuse. We'll use our fingertips so that it doesn't get too gory. But we'll just be attached by our fingers to the person next to each other, or to the cell next to each other. And that attachment is attached to the skeletal system. In this case, it kind of doesn't work really well. If we were perfectly round, then the analogy will make more sense, because like every single part of our body is attached to the skeletal system. But if we were perfectly round human beings, then, and if we just had like a weird like cross-like skeleton, then the attachment from me to the next human being would be at the portion where the skeleton, at the skeleton portion. That's kind of how cells connect to each other. They have anchoring points that are attached to the cytoskeleton of the cell itself. So these are like the strongest, or this is the most structurally sound portions of the cell, if you will. But anyway, these are desmosomes. This is a collection of proteins that allow cells to anchor to each other, or in the case of hemidesmosomes, that allow cells to anchor to the underlying tissue, in this case, your dermis. So in the case of keratinocytes, this is called stratum spinosum because the keratinocytes, they don't look perfectly round. They look like starfish or something along those lines. Something, they look spiny or so. And the reason being is because the cell loses its round configuration. It's almost as if you deflate the cell, but all of the attachments that that cell had remain. So so the attachments that, let's say if these were two perfectly round cells to each other and they form a desmosomal attachment right here, so they look like two perfectly round cells, no problem. But then you take away all of the air, you deflate them, but the connection is still there. So they start looking like only at the points of connections do they meet. So they start taking this spiny-like shape because of this. So that's the stratum spinosum takes its name for that. The cells are not perfectly round. They look like spiny and so forth. Stratum granulosum, keratinocytes, melanocytes. The way that the pigment is. So you see, let's go to a photo here. So you see, well, stratum granulosum, you're going to start seeing more pigment and those pigment granules, and also granules of keratin. They're going to give it this granular type of appearance. That's why it takes its name. And eventually, keratin, okay. Organisms. So this is what's happening at this level, the beginning of cell death, basically. Stratum lucidum, we mentioned, is only going to be present as a clear layer within the palms of your feet, hands and feet. And here's the stratum corneum, several layers of dead keratinocytes, water resistant but not waterproof. All right. So these are some of the specialized structures that I was referring to. Tactile discs. These are cells with sensory nerve endings. So these are the specialized cells that serve as an antenna function to translate sensory signal to the nerve endings for the cells that are, for the neurons that are found, whose cell bodies are found within the parallel to your spinal cord. So here's the cell body of one of my neurons, and that cell body sends a nerve, right, to every, like it sends a nerve to my fingertip. It's a long nerve. So that's one cell. That's why they say that I think you mentioned, I think you heard about neurons, that they have the possibility of being basically the length of your foot, because the cell body that innervates your toe, that's within the adjacent to this parallel to the spinal cord. So that's one contiguous cell that goes from your from the ganglia adjacent to the spinal cord to the tip of your toe. That's a long nerve, one cell sending a long axon, a long nerve. Now, once this nerve reaches the end of your toe, is just like any other axon, is going to have just free nerve endings present there. And here you have a Merkel cell that surrounds it. These Merkel cells create an environment that whenever there is, you sense that there is a touch, in this case in your toe, it's going to undergo a chemical reaction. In this case, it's going to release sodium, and that sodium is going to serve as a signal for this nerve ending to fire an action potential back to the sensory cell, and the sensory cell sending the signal to the neurons. These Merkel cells, these tactile cells, they are the ones that are kind of sensing the environment and then sending the signal to the nerve. Merkel cell with sensory nerve ending. They're like one kind of function found in hairless skin, respond to touch. Melanocytes, controlling melanin, that's good. So here is the reticular layer of the dermis. You're gonna have you're gonna have a lot of collagen and elastic fibers in different, without any regular pattern to it. What type of tissue does this? What type of tissue has lots of collagen without a regular pattern to it? Yeah. Lots of collagen gives it it's dense. The collagen is not regular, it's irregular. And whenever you think about fibers, you should immediately think connective tissue. Okay? Nerve is not reaching fibers. Muscles are not reaching collagen. They have their own fibers. Like actin is a fiber, but not these collagen fibers that we're. I guess whenever you see collagen, for the most part, you should think about fibers. All right, so here's the, so the dermis itself is classified into two layers, the reticular layer and this papillary layer. The papillary layer is not gonna be as rich in collagen, and that's areolar, it's not gonna be dense in collagen. It's going to have some collagen. And it's going to be areolar tissue. I gave it away. It's going to have some collagen, not that much. It's going to have fibers, elastic fibers, to produce some sort of to give it elasticity and so forth. But it's this is areolar tissue present here. Then you're going to have the reticular layer, which is going to contain dense, irregular connective tissue. Innervation, we have tactile corpuscles in dermal papillae. The pressure, that's going to be Pacinian corpuscles or lamellar. So one thing, these are very common names that they still persist within anatomy and physiology textbooks. One of the, I believe one of the goals of anatomy and physiology textbooks is to drive away from this naming nomenclature that relies on either who discovered it or who named a particular cell or structure. The reason being is because the name of the person doesn't tell you anything about the function. Meissner corpuscle doesn't tell you anything about the function, but if the name tactile corpuscle, that tells you that it has a sensory function. So whenever possible, I will just drive towards the the descriptive name rather than the name of the discoverer or the person that named it. So tactile corpuscle, you can hint that it has this tactile role, this sensory role. Lamellar corpuscle, we talked about these structures that translate the sensation of pressure. That it makes sense that a cell structure that has several layers to it, that it will sense pressure as if whenever you push one layer to the next or to the next, it will infer that as the quantity of pressure that is being applied. So these several layers, these several lamina, that the name lamellar should tell you that it has several layers. The corpa, and you just have to associate that corpuscles are these the name for these structures, these sensory structures that are adjacent to nerves to translate a sensory signal. Tactile corpuscles translate a touch signal to the nerve. Lamellar corpuscles, they translate the pressure signal to the nerve. These are not nerve cells. These are just cells that take the signal and send it to the nerve itself. They tell the nerve usually through a change in potential, a change in the environment around the nerve, either by increasing the amount of a particular ion surrounding the nerve. That's how they transmit their signal. Whenever we talked about, we will. I'm gonna save this for the nervous system. Whenever we talk about all the why oxygen is constantly required, it's because energy is constantly required. It's because we need to maintain these homeostatic mechanisms. One of the biggest consumers of energy that we really don't think much about is a mechanism that creates gradients of ions within the cells. Whenever you think about gradients, you should think about a battery. That's how a battery works, right? It creates a gradient of electrons, of ions, that whenever released, it will produce work. Similar to a dam, a water dam. It creates a gradient of water pressure driven by gravity, and the movement of this creates energy. Your cells create ion gradients. Basically, you can think about the membrane of the cell serving as a dam as well. And whenever you open this gradient, this movement creates, whenever you finally release the channels, whenever you finally open channels of sodium, these gradients these gradients get released, and in the process creating some sort of work, some sort of energy, an action potential, a signal between one cell and the next. Now, once you release those gradients, how you have to create the gradients once again. A lot of these gradients require active transport, so you're spending energy to create this gradient. Your sodium-potassium pump spends energy to create this gradient. You can think about every time there is an action potential, every time there is communication based between one cell and another, based on these gradients that you have basically expand the gradient, and now you have to recharge that gradient. Every cell has to recharge that gradient. It's like your phone, it needs to recharge. That recharge requires active transport. That active transport requires ATP. And that's why we require that's why we require constant energy. That's why we require we need oxygen to be present at all times. All right. Here you have the subcutaneous layers. Here you have these adipocytes, small nuclei, mostly vacuoles filled with lipids. What else do we have here? Here you have melanocytes with melanin pigment. Let's look at these melanocytes. So here you have melanocytes with which what they do is they release these vesicles. You see these vesicles, and those vesicles get endocytosed by adjacent cells. And those cells eventually, this is how this particular this particular endocyte. It's the membrane is degraded, and then the protein, the melanin, gets released within the cytoplasm. The melanin usually is there for protection of these nuclei, so the melanin is going to place itself on top of the nuclei in order to protect it. The difference in skin color is going to depend on number of melanocytes, quantity of melanocytes, and how active they are. We probably notice that they are more active in response to sunlight. You get exposed to sunlight, that activates melanocytes for them to release more melanin. More melanin, it's it's our way of tanning these fellas right here. All right. We talked about the role of your epidermis within the production of vitamin D3. The sunlight, we mentioned, is strong enough for us to feel the heat of it. That's energy that gets energy. That energy gets it gets transferred into cells within the surface of our skin, within our bodies. That transfer of energy excites bonds, electrons within our cells. That's conducive to movement. That movement is interpreted by nerves, by our sensory system as heat, and that's why we feel heat whenever we're adjacent to the sun. Now, that energy that's capable of producing all of these changes is also capable of activating this compound called cholecalciferol in the surface of our cells. The cholecalciferol, this is the initial compound, a sterol compound, that gets activated into cholecalciferol. Calciferol goes through an intermediate product in the liver, and eventually this stimulates this leads to the formation of calcitriol within the kidneys. This calcitriol is going to induce the the absorption of phosphate and calcium within the digestive system. So if we that would increase the amount of salt, of calcium and phosphate. Any deficiencies in dietary vitamin D or environmental vitamin D can lead to a decrease in the absorption of calcium by the digestive system. This decrease in the absorption of calcium, calcium is a main component of our bones, and this decrease in the absorption can lead to weaker bones, a condition called rickets. Is a consequence of deficiencies in vitamin D3. Deficiencies in vitamin D3 that lead to decreased absorption of calcium. Decreased absorption of calcium that leads to weaker bones, basically. It leads to decreased deposition of calcium within your bones. Whenever we get to the skeletal system, we will learn how dynamic bone calcium resorption and deposition is. This is a constant treadmill of depositing calcium and reabsorbing calcium. Calcium that is an ion that is necessary for several processes within our body. Calcium, whenever we can get it from our diets, from our environment, and we usually resorb it from our bones whenever it's needed. We need to constantly be have more calcium within our diet for this cycle to be at a homeostatic level. All right, we went over hair follicles, and here you have the sebaceous glands. Similar how we talked about how we, whenever we shower our skin, we wash away all of those oils that our skin naturally produces, and then we replenish them with either lotions or just time. Same goes for your hair. Whenever you shampoo it, you wash away those oils that are produced by these sebaceous glands that are part of this structure. We usually, once again, we wash it away and then we try to replenish it. Here we talked about, so let's go over some of the components of your hair follicles. You're going to have the connective tissue layer that surrounds the hair follicle. That's going to be what attaches the arrector pili. Next to it will be this glassy membrane. You will have the sheet of the, you will have a sheet that's surrounded. You have external root and internal root. And immediately next to it, you will have the cuticle of hair. You also have within the root itself, you will have a cortex and a medulla. The portion of hair that has proliferative, because your hair is constantly growing. Here you will have within the hair matrix, you will have this proliferative capacity that leads to the formation of the growth of hair. The hair papilla will contain the vascular components that, once again, they are there to supply these stem cells present here. Okay. During the lab, we'll go over some detail into these sebaceous glands. Usually, you will be able to tell the difference between different glands, whether they're mucus or whether they serve other roles. Whenever we were looking at the epithelium, we noticed that some epithelium will contain mucus cells. These mucus, goblet, clara cells, they would be clear. Same goes for mucus glands. They will be clear. Sebaceous glands are darker in nature, and so in this case, in histology, we'll go over them in more detail. But here, even here in this sebaceous gland, you will see that they are they're clear. They're comparable to mucus glands. But here, these are the glands, and they will be releasing their sebum, this oily, waxy substance within to the lumen that houses the hair follicle. Here, this is a cross section of a sweat gland. What type of epithelium do we have here? Anyone? Is it simple or stratified? That's the first question that you have to ask.
Simple?
Yes, it usually stratified is pretty obvious whenever it's stratified. If you see sometimes a simple tissue will look stratified, and that is because of the thickness of the section. So let's say that anyway, the thickness of the section, it's like if you see my fingers right here, you can tell that it's just one layer of fingers, right? But if you were to see them in kind of like an angle view, you will think that I have like 1, 2, 3, 4, 5 layers of fingers just like right next to each other, if these were layers. Same goes for these cross sections that we are observing. For the most part, they are one layer, but if it is a thick cross section, if it is we're looking at a tube, basically that's what we're looking at. And if that tube is thick and the layer and the cells are kind of overlapping, then this layer that's behind it would actually look like a second layer rather than being part of the same one. But yes, it is simple, and it's a squamous, columnar, or cuboidal.
It looks columnar, but it also a little bit cuboidal, so I'm confused.
Yeah, it looks like so in this case, for the most part, whenever we talk about glands, for the most part, they're going to be cuboidal. So just with that knowledge, I come already prepared with that knowledge. But now if you were to be given this and you had to make the decision, then I would go with I think in this case that informs most of the answer because this is the layer that looks cuboidal. And so based on this layer, it looks cuboidal. This looks cuboidal, this looks cuboidal. The reason why this layer doesn't look cuboidal is because I'm seeing two layers of cells overlapping each other, basically. this view here is the same situation, but it's harder to tell that it's 2 layers of cells. This could easily be confused as one long columnar cell. But hopefully you will see that there is a breakage at this level. There is a break in the cell layer that kind of hints that this is the end of this cell. And whatever we're seeing here, whatever we're seeing at the lumen level, that is just the cell that's behind it. It's just peeking over. So here we have sweat glands. We will go over this in a little more detail in the lab, but they look, once again, these are most likely simple cuboidal based on this, but they look like they are more, they have more, a more convoluted structure. All right, I'm going to just briefly go over the structure of the nail. Here you have the body, here you have the lunula, this area, white area that we all conceive. Here you have the nail root, and at the base of the nail root, that's wherever we're going to have this proliferative capacity. It undergoes this similar process that you can imagine your hair grows, your hair does, your epidermis do, your nails do. At some point they were cells, at some point they were living. But eventually they fill themselves with keratin, their organelles die, their nucleus dissolves, mitochondria, all of those dissolves, and they just become a big bag of keratin. And in this case, from the root onwards, it grows that way. The eponychium is this basically junction between the nail itself and the tissue layer. What do we have here? These cross sections gives us a view of the epidermis, the dermis. This is our bone, phalanx. Hyponychium is going to be kind of the distal junction between your nail and the tissue, the underlying tissue. So remember that. Eponychium and hyponychium. I guess you can kind of remember hyponychium. Whenever you think of hypo, is under, right? Under the nail. And whenever you think of epo or epi, that should be on top. So this hypo will be under, epi or epo on top. One last thing, we're going to look at repair to injury of the integument. So what's happening? You get a cut, bruise right there. This particular cut has gone through the epidermis and through the dermis, and it has kind of reached also the hypodermis here. What's going to happen? There's going to be an initial, all throughout there's going to be a migration phase in which just components of the epidermis, all of these basal cells that have this proliferative capacity are going to basically surround this tissue. You're going to have components of this original scab is going to be part of the, in this case, a blood vessel was ruptured. We mentioned this coagulation response, which we looked into detail moving forward. Here we see cells of the immune system going to clear these components. So this is the response to chlorine. And that chlorine is going to be surrounded by cells of the epidermis, basal cells of the epidermis. infiltration of macrophages and fibroblasts. Remember what fibroblasts do? Fibers, right? They make fibers. So they're the specialized cells, the core cells of the connective tissue, and they're responsible for creating the environment of the connective tissue, which is in this case full of fibers. So what's going to happen? These fibroblasts are going to basically they are responsible for creating the environment of connective tissue. They're doing so within this area. They're basically the connective tissue is regaining some of these lost ground. Fibroblasts are producing collagen, these elastic fibers, and we have this kind of like this contained healing process here, where the part that was originally clotted, coagulated, we consider that as part of the exterior environment. And these cells of the epidermis, these basal cells, these cells that they're growing and leaving behind collagen, and this will eventually, this coagulated part will become part of a scab that gets exposed, left to the exterior environment. This scar tissue, whenever we break this rupture, whenever we have a cut, we kind of break the kind of order of the collagen, the underlying collagen. We replace that with new collagen, but there's usually a scarring that occurs, a mark that is left behind. All right, let's leave it at that. We're gonna meet on Saturday for going over the integumentary system portion of the lab. Exam 1 is gonna be due by Sunday. Please keep in mind, reserve yourself some time for that. If you send me an email on Sunday at 10 a.m. with an emergency, I'm going to be sleeping. But don't worry. It's just like, talk to me. We can solve this. So if I don't answer immediately, don't worry. We can still solve it. We can solve it during our next class period. But make sure you talk to me. Talk to me in person rather than send me an email saying that this is about the problem. You can, whenever you talk to me in class, we can make sure that we solve it right then and there. Okay, so join classes. I'm okay with this cadence because I know that you are, that some of you are looking into the videos. But once again, participation is something that we're going to take into account throughout the semester. There's no grade for it. However, at the end of the semester, if you're borderline, then that's whenever it comes into play. So keep that in mind. All right, everybody. Thanks for joining, and I hope you enjoy the rest of your day.